Hi all, I'm Aisha Lawal, and today I'm going to be giving a short presentation on a project I've been working on. And the title of my talk is HyperPredict Estimating Hyperparameter Effects for Instance-Specific Regularization in Deformable Image Registration. So I've sort of highlighted deformable image registration here in red because that's the main focus of my research. But this particular project is looking at a way to make the process of image registration more efficient. So just as a side note, this work has been a collaborative effort with my supervisor, Ivor Simpson. We're both from the Predictive Analytics Lab, and we're both members of the Sussex AI Research Group. And as a domain overview, I'm working with medical images, so this can be applied to data sets acquired from different parts of the body, which includes the brain, the abdomen, the spinal cord, amongst others, and also data sets acquired from different types of sensors, right? So what's image registration? In simple terms, it's basically an alignment problem. So technically, this means that given one image, which we call the moving image, and another which is termed as the fixed image, what we try to do is we try to find an optimal transformation T that moves or transforms the moving image to the coordinate space of that of the fixed image. But why is this an interesting problem? There's a large set of possible clinical applications where image registration is used in. And one of them is segmentation propagation. That's basically when you try to like transform or transfer segmentations from the space of one image to the space of the other. And another is when you're working with, when you're trying to do a longitudinal analysis. So for instance, that's looking at the cortical development of the human brain. When you acquire the a particular scan of a particular patient at different time points, and then you try to analyze the cortical development of the brain for that particular patient. And another is when you're dealing with computational anatomy, that's basically disease progression for a period of time. So like every other optimization problem, we have an objective, right? So with image registration, we actually have two competing objectives. One is a similarity term that tries to match the fixed image to the moved image. Yeah. And this can be your mean squared error, normalized cross correlation, or mutual information. Selection of this metric depends on the modality of data they are dealing with. Yeah. But the problem with this is the problem with optimizing for just this similarity this similarity term is that it's ill-posed because we have a large set of possible solutions. So we need to somehow constrain this. And we do this by introducing some form of regularization. So this means basically that we have a large set of possible solutions, but we're only interested in those that are plausible. So how well do we know how do we know how well our image has been registered? So we can look at the anatomical overlap. That's we acquire labeled we label the fixed and the moving images. We pass the moving label through the deformation field. So we transform the moving label with the deformation field that has been acquired from the registration method, and we derive the moved label, right? We can then compute the dice score between the fixed label and the moved label, which is basically a measure of overlap of the anatomical structures. So ideally, what we would want is a high dice score, that's a dice score closer to 1, because it means that the anatomical structures do overlap. And if it's closer to 0, it means that the anatomical structures between both images do not overlap. Another measure of evaluation is by looking at how smooth the deformation field, and we can use the Jacobian determinant to tell us how smooth it is. So if we have the Jacobian determinant greater than 1, it means we have like a local expansion within that region. If it's less than 1, it means there's been some squeezing or contraction within that region. And if it's negative, which typically we try to avoid, it means that there's been some folding within that local region. So if we try to sort of understand the effects of regularization, which is basically the lambda highlighted here in red, you see that visually it's a bit difficult for you to see. These are registered images, right? And visually it's a bit difficult for you to see the difference between when no regularization is applied at all and when you apply some form of regularization, that's when the value of lambda here is greater than 0. But if we examine the deformation field, you see that with no regularization at all, we have that there's some form of folding within this region, right? And if we apply some form of regularization, we have a smoother deformation field, which typically is what we try to aim for. And recent work has actually shown that this value of lambda does vary per subject, and it also varies per anatomical region. So if you're looking at trying to optimize for the hippocampus, you have a different optimal value of lambda compared to when you're trying to optimize for the ventricle or the putamen. So this means that what's the optimal value of lambda? Or how do we find the optimal value of lambda for each task? So ideally, what we want is that we want to use instance-specific values of lambda based on the task that we're dealing with, or what it is that we're trying to optimize for. So we have to look for the optimal value of lambda for each instance. Traditional methods of doing this is by using cross-validation, where we get a validation data set. We discretely sample values of lambda, we run it through the validation data set, and we select just a single value of lambda that is optimal, and we use that across the entire test data set. But then this is suboptimal because, as we've seen in the previous slide, the optimal value of lambda does vary per anatomical region, right? So we can't just use one across the entire test set. We get it results in obtaining suboptimal results. So in addition to that, it's computationally intensive because we have to run multiple times. And also we're limiting the search space because we're discretely sampling values of lambda. So with HyperPredict, what we're proposing is, can we sort of establish a relationship between the moving image, the fixed image, and a continuous range of hyperparameter values with the evaluation matrix that we've seen before? So we've seen that we can analyze or look at how well our image has been registered by looking at these two metrics, which is the dye score and the number of folded voxels. So can we pass in these as inputs and sort of predict what the associated dye score and the number of folded voxels are? So one important point to actually note is that we predict 35 different dye scores. That's one for each anatomical structure. And that's because we're trying to select the optimal value of lambda per anatomical region. So this enables us to do that selection. Optimization process is done by obtaining ground truths from we used two different registration methods. One was NiftyReg, which is an algorithmic approach, and another was CLAP-IRON, which is a deep learning method. So we obtained the ground truth for both the dye score obtained from the labels and also the number of folded voxels obtained from the deformation field, and we optimize based on that. So this means at test time, we do not need to run multiple registrations in search for an optimal hyperparameter value. We can just pass in our moving and fixed image and a continuous range of hyperparameter values through this MLP, and it predicts the associated dye scores and number of folded voxels. Then based on that, we then select the optimal value of lambda. We pass it through our registration algorithm, and then we obtain our deformation field. One thing to actually notice is with our method, we're able to define a flexible selection criteria of these values of lambda. So with this, basically what we said is we do not want, because we're testing thousands of hyperparameter values from a continuous range, we do not want, we can say we don't want values of lambda that lead to us having the number of folded voxels being less than a certain percentage, right? Being greater than a certain percentage. So in our case, we said we're not interested, or we discard values of lambda that leads us to have the number of folded voxels greater than 0.5%. So this, with our method, we can sort of define this criteria at test time. So we do the selection of the optimal value of lambda and pass it through the registration method to obtain the deformation field. And we see that with HyperPredict, if we try to analyze the predictive abilities to see how well we're able to predict the associated matrix, ideally with a Blunt-Altman plot, we want our points to be within the region of the mean, which we see in both of them for both the dye scores and the number of folded voxels. And if we compare this to cross-validation, although we have like comparable dye scores with, I think, the amygdala and the pallidum, HyperPredict performs slightly better. But one interesting thing to note is that we have less folded voxels with our method. So with HyperPredict, we have more plausible solutions compared to with cross-validation, which is something that we want. And also because we're able to predict 35 different dye scores, it means we can select the optimal value per anatomical region. And then we use that to optimize based on what region we're trying to optimize for. So here we're trying to optimize for the thalamus, and the optimal value of thalamus here is 0.09. So we use that for registration and we optimize for the thalamus. Similar to that, the optimal value of amygdala is 0.16, and we use that for registration to optimize for the amygdala. And an interesting point is that we have, with our method, because we're now running multiple registrations in search for an optimal hyperparameter, we have that we can test thousands and thousands of hyperparameter value without worrying about computational burden, right? So in this case, we tested 8,000 hyperparameter values in just 0.84 seconds. That's without registration. So when we then select the optimal value from this, we can then register it using any registration method with the selected optimal value of lambda. We also find actually in our paper, we performed some ablation studies and where we tested 40,000 values within a short period of time also. So the computational efficiency at test time is an added advantage of using HyperPredict. So in conclusion, basically, we have shown that we can sort of establish a relationship between the image pairs and a continuous range of hyperparameter values, lambda, with the associated evaluation matrix. And we also show that we've seen that the optimal value of lambda does vary per subject anatomical region. So ideally, we would want to select instance-specific values of lambda for each, and based on what it is we're trying to optimize for. And at test time, we have a flexible selection criteria that we can tweak based on our preference. So thank you so much for listening, and feel free to scan the QR code to read more about the paper. Thank you.